Welcome to clickmyproject.com. Now I am going to explain the quantitative assessment of diabetic macular edema using fluorescent leakage maps. Now I am going to explain the concept of this process. Here we propose a quantitative assessment of diabetic macular edema, that is DME from fundus image sequences. So for that we present a method for processing fundus images to generate a fluorescent leakage map that can be used for quantitative gauging of DME. Model the spatial distribution of the image intensity within the macula is the initial process in our method. Shape features play an important role in computer vision, image processing and pattern recognition. We introduce a novel method of using SVM, that is support vector machine classifier, and shape feature. Finally, deduct the normal or abnormal image from the fundus image. This model, which represents the non-leaking structures, is then subtracted from the late time frame image to enhance the areas of the fundus image. This is the flow diagram for this process. First, collect the fundus images and store in the database. The next step is the RGB color separation. In this process, the RGB color model is an additive color model in which red, green and blue light are added together in various ways to reproduce a broad array of colors. The next process is the pre-processing step. In pre-processing method, median filter is used to remove noise from the input images. It is often desirable to be able to perform some kind of noise reduction on an image or signal. The median filter is a non-linear digital filtering technique often used to remove noise. Such noise reduction is a typical pre-processing step to improve the results of later processing. Median filtering is very widely used in the digital image processing. Then the next step is the vessel segmentation. For vessel segmentation, here we use graph tracing algorithm. The vessel will be segmented in the image by the use of graph tracing algorithm. Each vessel is tracked individually by repeatedly finding the next vessel point with a scoring function that considers the pixel intensity and orientation in the vicinity of the current point in the image. Tracking for the same vessel then continues along the most likely path. Next, we model the segments as a segment graph and use constraint optimizations to search for the best set of vessel trees in the graph. Then the next method is the feature extraction. For feature extraction, here we use shape feature extraction method. For shape, we consider two methods. First one is the moment invariant feature extraction. It's a function of image moments. Moments are the projection of the image function into a polynomial basis. Moment invariants have been frequently used as a features for image processing, remote sensing, shape recognition and classification. Moments can provide characteristics of an object that uniquely represent its shape. Then the next thing is the Gabor feature extraction method. For Gabor feature extraction, as the core of the Gabor filter based feature extraction is the 2D Gabor filter function. Gabor filter is a complex plane wave multiplied by an origin centered Gaussian along with the Gaussian linear axis and gamma sharpness along the minute axis. Then the final step is classification. In classification, we analyze the fundus images, normal image or abnormal image. For classification, SPM classifier is applied in our process. SPM means support vector machine. Machine learning is a field of study that gives the computers the ability to learn without being explicitly programmed. And machine learning algorithm like SPM, ANN is used for any processes. Then the Classification process is data step. Our data set contains the classes, each samples. The data is said to be plot original data for visual inspection. Then the last step is the performance analysis. For finding the better performance, we have to find the accuracy of the process. Accuracy is mainly based on the classification process. This is the MACULAR command window. These are the function files used for this process. Main is the main function file and the others are the sub-function files. Click the main function file and right click, choose the run file option. This is the design guide window for the identification of diabetic macular edema by using fundus image. First, input image button is click and load and choose any one of the image. The next one is the RGB color separation. In this red channel, green channel and blue channel is individually separated and displayed in the figure file. The next process is pre-processing step. In pre-processing step, the noises are removed and the filtered image is displayed in the denoised image button. Then the next one is the vessel segmentation process. In this vessel is that is blood vessel is segmented, extracted from the fundus image. Then also calculate the blood vessel thickness value. The next one is the feature extraction. Here we calculate the feature extraction method is applied for both the training image as well as the test image. So the testing image values are displayed separated, then training images are displayed separated in the table file. Then the next process is the classification. In classification, SVM classifier is applied, then calculate if whether the input image is the normal image or the abnormal image. Then finally, find the performance analysis. Here we plot the graph for the accuracy value and sensitivity factor and also specificity factor. Here we see the values for the accuracy. Accuracy we get 97.5 and also sensitivity and specificity factor. Finally, we conclude that the quantitative assessment of fundus image has been proposed. Classification system also based on the evaluations and measurements. Thank you.